My name is Mark Tefero Chivumbi, and I'm going to take you through a publication I happen to do with Associate Professor Claire J. Stanley from Georgetown University. She's based in Germany, and this is a systematic review we did on antimicrobial resistance in Uganda, and we happen to publish this in MDPI. It is open access, and this is how it looks like when you open it in MDPI. And this work was actually accepted in May 2021, and so that is how new the work is. So I'll go into the presentation right away. So firstly, I'll just tell you briefly about the paper, and then I'll also tell you about systematic review of the process, how it is done. So that was the background of the paper. It was about the global burden of antimicrobial resistance. And we discovered that a multidisciplinary collaborative regulatory approach is needed to combat antimicrobial resistance since it spreads in the environment, and development of resistant microbes is a challenge to control antimicrobial resistance. As I've told you earlier, it was a systematic review, and unfortunately, we only used one database because there were only two. That was me and my co-author. So we only used PubMed for this case, and through PubMed, we collected 427 papers. I'll show you the research syntax we used at the end of the presentation. So we were able to get 4 to 7 hits from PubMed, and we got 3 other papers from Gray Literature, and we had a total of 430 records. So after reviewing the records through the abstracts, through our rejection criteria, we were able to exclude 264 records that were irrelevant to what we wanted, because we're looking at papers that were on combating antimicrobial resistance in Uganda. Some of these papers were about antimicrobial resistance combating, but not in Uganda. Some were not about antimicrobial resistance, but were from Uganda, while some were papers that were about Uganda, but did not have sufficient data to qualify as combating antimicrobial resistance in Uganda. But rather, some just mentioned it, and they happened to appear as research for papers. So we zeroed down on 166 full articles, and we scrutinized the full articles. We read these papers from the topic up to the last word of each of these 166 papers, and we were able to exclude 3 papers after discovering from their full reading that they were not actually relevant to what we wanted. So in the analysis, we actually used 163 papers in the analysis. So a sneak peek into the results. This is a picture of Uganda showing the geographical distribution of the studies. And as you can see, this image was generated using Mapbox. It's a software that is used for generating maps with distribution of data. Unfortunately, I didn't do this. It was done by my co-author, Dr. Claire. And so she generated this. This is not a free software, so you might require a license to use it. Yeah, but it comes in very handy when you're dealing with a graphical representation of data on maps. And then a sneak peek into the results. From our analysis, we generated this output of our results, and we categorized the output in terms of antimicrobial resistance. In terms of antibacterial resistance, antimalarial, antiviral, and antifungal. And so we use the matrix. And on this upper hand, as you can see, we had MR context, study design, and output. which were further subdivided into human, veterinary, One Health, and under the field study, we had fields under study designs. Some were done in the field, while some were basically lab studies done on archived samples. And in terms of output, we're considering whether the paper was on surveillance of susceptibility or resistance genes. And we also included policy, but we actually discovered that there was no paper that was actually on MRA policies in Uganda. So that was the matrix of the data that we came up with. Then we further went on to generate a graphical representation of the data we collected. And as you can see, this graph was actually generated using R. And from this previous table, this work was done in Linux. So we basically just used Linux and R for the whole analysis of the data. We didn't use Excel, we didn't use SPSS or Stata. Yeah, we just basically used R and Linux since it was what we were comfortable with and it was easier to use. So this graphical representation, here on the x-axis, we had publication years, and on the y-axis, we had frequency, and the different colors as per the legend show the different papers, whether the paper was on antiviral resistance, anti-malarial, antifungal, or antibacterial. And as you can notice, there were very few papers on antifungal resistance and also little studies on antiviral resistance, while anti-malarial alone had a lot of work done on it. And there was a general rise in papers on antimicrobial resistance in Uganda. So one thing to note from this previous table also was that One Health studies were few, veterinary studies were also few, but more than One Health studies, and human studies were very many. And as you can see from the first column as well, most papers were on antibacterial or antimalarials, and fewer on antivirals and antifungals. So from the other part of the results, we also analyzed the common resistance genes that were identified from the studies. And as you can see, E. coli and Staphylococcus had a good number of studies done on their resistance genes in Uganda. And we also had other studies on Streptococcus, Salmonella, Mycobacterium, Klebsiella, and Enterococcus species. Antimalaria alone had over 55 studies on resistance genes of malaria. And I think most of the studies were actually on malaria in the earlier days in Uganda. That's why it happened to have a very huge number of studies, even when it comes to resistance genes. And antivirals, we had a few papers on hepatitis and HIV in terms of their resistance genes in Uganda. So in conclusion, we proposed a One Health approach would be better to be used in the National Action Plan to combat antimicrobial resistance. And as you can see, the Uganda National Action Plan in 2017 actually highlighted this, but we are yet to see if it is being implemented. And also, a multidisciplinary task force was created in 2017, and it's led by the following institutions, and it's funded by the UK. And these are the recommendations of the study. And these were the partners of the, these are my university that I went to, and my co-author was from Georgetown University. Then I'll probably take you through of how we were able to do a systematic review and make this work reproducible in case someone wanted to actually try and do what we did from our study. So I'll stop sharing this, and I'll probably share the page where the paper is located. so that you can see how it looks like.
Thank you so much, Mark, for that wonderful presentation. So if you have any questions, sorry.
I wanted to show just how the paper looks like and where we added extra work in case one wanted to redo the experiment we carried out. And so you're seeing my screen. This is how the paper looks like. And when you look down here, there is a section of supplementary material. So in that section, we added the search syntax that we used and the papers that we actually included, including the full texts and everything about that we used to consider the paper, whether it fitted our research criteria or it didn't. So I'll stop sharing this. So that is the paper. It was published in MDPI. I'll share the link for the paper in the chat.
Thank you so much, Mark. So there's a question in the chat, and anybody else can please feel free to also share a question. So Benson says, what tools did you use to find the papers?
This was a systematic review, and I initially wanted to get papers from all databases, but were only two. And when we used PubMed alone, we discovered that we had over 420 papers that we had to look into. And since we were few, we zeroed down on PubMed because it is a public database and it's free of charge, because the other databases that have studies on antimicrobial resistance, some are public, some are private, some are non-profit, and we also had other databases we had zeroed on, like Web Science, Google Scholar, but we didn't use these because now, for instance, some papers in Google Scholar and some papers in PubMed would actually be the same. That means we'd be having now more work of having to get duplicates from Google Scholar and from PubMed. So, but for this study, we only used the papers from PubMed as our database.
Thank you. Is there any other question? Okay, I hope I'm not closing anybody out, but I'm sure Mark will be out. Sorry?
Maybe what I can do, yeah. The person who asked how we got the papers, maybe I can open my PubMed and just show them how to use the advanced search syntax, but not actually do the search itself.
Sorry, what did you say?
The person who asked how we got the papers, I don't know if I can just open PubMed shortly and just show them how to use the advanced search syntax in PubMed.
Okay, I think you can just share a link, right, to the chat. to PubMed.
Okay.
Okay, thank you. Okay. So our next speaker is here with us. So we have Trisha Shah will be going next as the first keynote of the day. Just a bit about Trushar as he shares his screen. Trushar Shah is the Integrated Breeding Platform Hub Manager. He is a Kenyan national. He graduated from the University of Bristol in the UK with an honors degree in biochemistry, molecular biology, and biotechnology. He holds an MSc in molecular modeling and bioinformatics from Birkbeck in the University of London. Welcome, Trushar.
Hi, thanks very much. Can you hear me?
Yes.
And you can see my screen?
Yes.
I hope it's in presentation mode. Okay, super. Thanks very much. Thanks, Margaret, for that kind introduction. And thank you to all the organizers of this conference for the invitation to speak to the whole group about plant genomics. So I've titled my talk called Genomics Going Bananas, and hopefully by the end of the talk, you'll see why. So I'll be presenting a kind of overview in the banana world of how genomics has helped in the efficiency in our breeding program, and also to help speed up our breeding activities in the banana breeding setup here at IITA. So I work with IITA. IITA is the International Institute for Tropical Agriculture, where I'm based here in Nairobi, Kenya, but our headquarters is actually in Ibadan in Nigeria. And we work on a number of crops, and one of our mandate crops is actually bananas. And so I'll be talking to you a lot about the progress of genomics in bananas and how we're applying it to basically increase our genetic gain in the breeding programs. So just a little bit of background. So hold on, let me just get a pointer because I may need. Okay, no worries. Okay, so just a little bit of background about bananas. I mean, I'm sure all of you have had bananas. It's a member of the genus Musa, and as you know, there are many different types of bananas. There's bananas for cooking, a staple in a number of countries. There's a banana beer and juice. There's bananas for dessert, which are the sweeter bananas, the sukari ndizi, and the ones we get here in Kenya. They're also actually ornamental bananas, and in a lot of places they use the leaves and the fiber coming from bananas for different commercial products. So if you look at the picture on the right here, it just gives you the story about the domestication of bananas, basically how bananas go from the wild relatives, what we're calling the WR over here, which is kind of, hi, can you hear me?
We are seeing the first slide, maybe.
Oh.
Yeah.
Hold on. Do you see it now?
No, just the topic plant genomics.
Oh, okay. On my screen, I see it. Let me just stop sharing and reshare.
Yeah.
Sorry.
Okay. Yes, now we see the next slide.
Can you see it now? Okay. So sorry, I was referring to this picture over here about the domestication of bananas. So basically what you see on the right, are you able to see my pointer? Yeah, okay, great. So what you see on the right here is kind of wild bananas, which had basically a lot of seed and very little pulp, the fruit pulp. And then the current domesticated bananas that you all are pretty much familiar with, is basically the inverse of that. So more pulp, greater fruit filling, what we call, and less seeds, or in fact no seeds in a lot of the triploid bananas and even if there are seeds, those seeds are sterile. So that's kind of one of the interesting things about bananas is the whole domestication. At IITA, we're focused more on the breeding of bananas. So we want to be looking at different traits in bananas, which we would like to improve. Obviously, there's a high yield gap between performance and the research fields and performance on the farms in terms of the yield. There are also a number of major constraints in terms of pests, diseases, and I will cover a few of those in the next slide. And it's actually a very, it's a staple food crop in a number of countries, especially in Uganda, where, there's a high per capita consumption of the cooking bananas, matokes, where they consume close to 400 to 600 kg per person per year in Uganda. So we have colleagues also in the CGIAR, colleagues at Bioversity and the CIRAD, the French research agency in agriculture, who are more interested in kind of the evolution of bananas, where they came from, how the domestication happened, and things like that. So there's a whole kind of genomics aspect related to the domestication, because these are interspecific or interspecies crosses that have happened. And I'll talk a little bit more about the ploidy as well in subsequent slides, but you will see that they have a range of ploidies from diploids to triploids to tetraploids. And so the group in CIRAD and Bioversity, the Alliance Bioversity, they are focused more on aspects of evolution and domestication, whereas at IITA, we are focused more on the breeding aspect. So how to improve these bananas for smallholder farmers. So this slide just shows you some of the major constraints. There are lots of diseases, and these are bacterial, they're fungal, and also pests like weevils and nematodes as well. So these are some of the major constraints that our breeding project is trying to address in terms of resistance or tolerance to some of these pests and diseases, and also increasing tolerance to drought and a number of threats. You may have heard recently of a major threat to bananas, which is the Panama disease, the tropical race 4 of the Panama wilt disease, which is wiping out a lot of monoculture plantations in Asia, and is already in Africa. And so we're also looking at breeding towards resistance for these tropical race 4 Panama wilt disease. So in terms of what the breeding work involves at IITA, we basically breed for these three types of, the 2 types of highland bananas, what we call the Mushare, which is basically comprises of an AA genome, A standing in this case in terms of the nomenclature for the Acuminata genome, and the B, which you get in plantains, is the Balbisiana genome. So you have diploids, which are Mushares, and we're doing the breeding work for this in Arusha in Tanzania on the slopes of Kilimanjaro, and the matokes, which are the AAAs, which is the predominant sort of cooking variety in Uganda. And we're doing the breeding work for that in Sendusu and Namulonge, together with the national partners NARO in Uganda. And in Nigeria, we have a plantain breeding program. And these you're able to see here, which are basically comprised of, it's a triploid genome comprising of 2 A's and a B. And so we have breeding work going on all of these different types of bananas. So when you look at conventional banana breeding, it's actually a very slow and very expensive process. You basically start off by crossing. Over here, you see there's 2 eggs. This is coming from a wild type banana, which you see the fruits are not very, very filled. You cross that with a 3X, or it's kind of what you like. And then you will get from that, you will get a range of different ploidy levels because of the way the chromosomal or the meiosis takes place. And you'll get a range of them. And what we then do is we then take the 4X arising from the 3X by 2X. So there's a 4X that comes out of there. We then cross that again with an improved 2X now. So remember, first we used the wild. Now we have an improved 2X over here. And then from that, we get the 3X bananas. And remember, these are the 3X are actually sterile. So the seeds do not, they do not have seeds. And if they do, then they're sterile. So they're not able to germinate. So this whole process can take conventionally from up to 10 to 15 years from the time we make the initial process to the time that a banana variety is released. So it's actually a very slow and expensive process. And so what makes it slow and expensive is obviously, you saw we play around with different ploidy levels. There's the aspect of parthenocalpy, which basically means it doesn't set seed. The seed production, especially for 3X, is very unpredictable. You could get crosses where you could only get 1 seed at the end of it, or you could get a cross, depending on what 2 parents you cross, you could get thousands of seeds. And so even if you do get seed, there is a the germination rate is very low. So the seeds to embryo germination ratio is very, very small. And that's why we go through a step of tissue culture. And so basically, once you harvest things from the field, you would then have to go through a step of tissue culture into the screenhouse. Once the tissue culture plants come out, you have to harden them in the screenhouse and then take them back to the field. So this also adds to the length of time that it takes to get from a seed to a plant in the field. This process alone could be take almost a year. And then once you grow them, then depending on when they flower and also when they start bearing the fruit, so when the bunches are harvested, the first selections are done after 1 to 2 years. So because it's in the field a long time. And one of the things is that about 99.9% of the hybrids in the early evaluation trials are actually discarded because we get a lot of sort of linkage from the wild material, and the fruit filling in a lot of these is very, very low. So obviously one of the easy targets for us in terms of genomics work would be targeting this fruit filling trait, and that's something that we already are doing. And the other thing, to replicate any plants, to have a proper kind of experimental design in terms of breeding, it takes a lot of space as well, because one plant, you can imagine, takes almost 6 meters squared. So you need sort of hectares of land to do a sort of any decent kind of experiment, or even grow out a large population if we're to do the kind of work that I'm talking about in the next few slides. So I've told you how long this breeding takes. So this is where the genomics part of it comes in. So, can genomics come to the rescue? And, as new methods are developing, we have had sort of the first draft genome of banana, which was a double haploid Bahang. So double haploid because at that point, it was the draft genome was done through a combination of sort of Sanger sequence reads and using the Roche 454 technologies. And then in 2016, that was improved. That version 1, the draft genome, was then improved by adding sort of Illumina NGS reads onto there. And then in 2021, there's been the version 4 now, where we've basically got using the nanopore sequencing, end-to-end sort of gapless sequence of the whole genome for one of the species here, Musa acuminata. We also have the Musa balbisiana genome available, and a number of other genomes of varying sort of quality available. And all these genomes, if you're interested in them, are available on this particular site, the Banana Genome Hub. This is a site hosted in France with the colleagues from CIRAD and Bioversity have developed this kind of platform. You're able to do a lot of things on this platform. You're able to sort of visualize genomes. You're able to search for genes, or do sort of conversion from different versions of the genome. So if you have developed any kind of markers on a particular version and you want to see where those fall on the newer versions, you're able to do all those kinds of analysis on this platform. So, just making this available openly, all the sequence data and the resulting sort of analysis of marker information has really helped to accelerate some of the breeding activities. And I'll go through a few of those in the next few slides. So this is where genomics can come to the rescue. You must have heard the term coined genomics assisted breeding. And so in genomics assisted breeding, basically what we're trying to do is, let me just move this, is basically what we're trying to do is speed up the selection. And to do this, there are a number of different approaches. We've started doing some for the major constraints that I mentioned. We've started doing QTL analysis, basically in diploid. And now we're moving to more polyploid populations. So we've got populations which are contrasting for things like Fusarium wilt, for weevils, and for nematodes. And later on, I'll show you an example of what we've done for the weevil population. We're also looking at genomic selection, where it's basically used more for sort of quantitative traits. By quantitative traits here, we mean sort of traits controlled by multiple genes or multiple loci. And these include things like yield and other sort of agronomic traits. And then we've also done some genome-wide association studies for things like drought and parthenocalpy. And this is done basically while this is done in sort of structured populations, the GWAS is done in sort of diverse populations coming from very diverse backgrounds. And then we look at different marker platforms. I'll talk a little bit about a few of these marker platforms that we've used to speed up these kind of selections. Other technologies we've used transcriptome studies, so RNA-seq, looking at gene expression, particularly for disease. I'll take you through an example of that. There's a large genomics effort where we're looking at sequencing of matoke and plantain breeding material. So 100 accession of each, and this was being done through the African Orphan Crops Consortium, based out of ICRAF here in Nairobi. And then other areas where we're interested in, which I won't cover today, is things like looking at flower biology, because I said, seed set and all that, parthenocalpy, all relating to kind of the flower biology. So here we could look at, single cell sequencing of pollen and get a better idea of that. And also issues around chromosome doubling and heterosis, meaning, where you get a sort of additional impact from making your crosses based on coming up from different sort of heterotic pools. And what we're trying to do is also look at the whole diversity in our breeding program and divide that up to exploit heterosis better. So one of the major diseases, as I've mentioned, is banana xanthomonas wilt. It's basically causes wilting of plants. You can see here a healthy plant and then the wilted plant over here. And basically, because of the premature ripening of fruits, you don't even get a proper bunch. And eventually it results in the death of the whole plant. And so, unlike other banana diseases, which cause sort of incremental loss over years, remember the banana plants are in the field over a long period. The impacts of BXW are both very rapid and very extreme. So you get kind of extreme losses over here. So one of the studies we carried out was basically to look at BXW, and we looked at a transcriptome study here. And in terms of the experimental design, we had two different genotypes, one which was resistant to BXW, coming from the Musa balbisiana, and then the BXW susceptible banana genotype called Pisangawak. We looked at it at two different time points. And obviously we had, since in RNA-seq studies, the replications, biological replications are important, we looked at 3 reps of it each. We also carried out artificial inoculation with inoculum of the Xanthomonas campestris pathogen. And for the sort of control, we inoculated with water, just so that we can eliminate what we call the wounding effect. So just, you know, by inoculating, you might be altering the transcriptome of these just because of the wounding effect. And so we wanted to wound both the control as well as the treated samples over here. So basically we had 2 genotypes, 2 time points, 2 treatments, and 3 reps each. So as we went through this, what we did was extracted total RNA using the QIAGEN kits, and then the quality of this RNA was assessed and quantified using nanodrop. We prepared the libraries, cDNA libraries, and then we went for paired end Illumina sequencing using the HiSeq, generating reads of about 100 base pairs. So in total, what we got is about 559 million reads that were generated. And all this data set is available in the short read archive, and that's the project code over there if any of you is interested in looking it up. So in terms of RNA-seq, what happens is you get your RNA samples, you do the kind of quality validation, which we did using the nanodrop, and the output you will get from your Illumina HiSeq is basically these raw FASTQ files. So from the raw FASTQ files, you obviously generate some kind of QC reports just to see that all the kind of QC parameters, looking at you know, what your adapter sequences are, how many reads you have, what are the distribution of these reads in terms of read lengths, quality scores, all of that. So you need to do some kind of QC. We then, once we've done the QC, we then look at trimming off all the adapter sequences. So we then call that the filtered FASTQ. We take the filtered FASTQ and map, in our case, to the reference genome. We did a lot of this work on version 2 of the double haploid Pahang genome, which is the A genome basically. And you could use different aligners to do that. You map your reads, and then what you generate is basically the number of read counts per gene. And then you use different toolkits. You could use things like DE-seq or EdgeR. These are packages in R, which you can then look at what your differentially expressed genes are. So in our case, these were the tools we used. We used Trimomatic for trimming all the reads. We used FastQC for the quality control. Alignments, we used HiSat2. And then we selected the reference genome based on Busco kind of analysis in terms of completeness of the genome. And so we selected this reference genome out of all the ones listed in the Banana Genome Hub. And then we used HTC count to count the number of reads and mapped and annotated those genes. So this is kind of the distribution of what we saw at different, in the different genotypes and different time points. So P here for Pisangawak and M for the Balbisiana, 12 hours post-infection and 48 hours post-infection. And this is just sort of showing you the number of differentially expressed genes, both in terms of downregulation and upregulation. We then went ahead and mapped all these genes using a software tool called MapMan. We used a sort of ontology-based mapping to map all these differentially expressed genes onto particularly this biotic stress pathways over here. And you could see here, so this is basically your Balbisiana, this is your Pisangawak. You can see that 12 hours post-infection in the Balbisiana, there's kind of a burst of activity with a lot of genes being up and down regulated. And you see less of that in the Pisangawak, which is the sort of susceptible genotype. And then 48 hours post-infection, you see that it kind of calms down in terms of differential gene expression. So based on this work over here and based on the mapping, we then identified some candidate genes, and we then also validated those candidate genes using QRTPCR. So we took a subset of about 30 genes, and you can see here the trend for, in green, the RNA-seq, in orange, the QRTPCR. And you can see that wherever we did see some upregulation or downregulation of these particular genes, and these are the genes listed over here, that the QRTPCR also validated and had the same trend over here in terms of up or down regulation. The fold change may be slightly different, but at least it was correlated with the sort of expression patterns we were seeing. So one of the genes we identified from that was actually was an ortholog of a gene known in Arabidopsis called DMR6. DMR stands for downy mildew resistance. And so we identified that ortholog coming from the transcriptome work that we did, the RNA-seq data. We looked for it in terms of orthology, using some comparative genomics tools. So we went into the databases and looked for all the sort of orthologs coming from Arabidopsis, from Tobacco nicotiana, and also the other related Musa species of Albiciana over here. And from that, we could see that it fell in the same clade from the resistance gene identified in Arabidopsis as well. So we picked one of these candidates. We then went through and with our genome editing platform here, using CRISPR-Cas, we then edited that gene over here, the DMR6 gene, and you can see here the guide RNAs that we designed, the PAM sequences, and basically what happened is we got an 87 nucleotide deletion of this region here on this last exon of that gene. This is the Musa acuminata DMR6 gene. The last exon here, we got an 87 nucleotide deletion. And so we then looked at these genomic, the genome edited events over here to see obviously in the gels, and then also in terms of the plants. And here you can see the edited versus the control, and you can see the level of resistance that it conferred to BXW in this case. So that's kind of a nice example of how genomics has helped in identifying sort of potential solutions in terms of genome editing for some of the major constraints that we are facing. We've also been doing a lot of QTL mapping. One of the ones, remember I mentioned one of the constraints was weevils. And we did this basically in a triploid banana population. So we had two parents, the Moniette, which is a 4X, and the Kokopo, which is a 2X. We crossed these two, and most of the progeny we got was the 3X. We then went ahead and did the QTL mapping using this approach called continuous mapping, basically, which takes in this case, we used the DART-seq data. But here, what we did is rather than using the output coming from DART-seq directly, we asked them actually for the raw sequence data, and then we mapped and created our own pipeline of analysis to do the dosage calling basically as triploids and tetraploids in these cases. So instead of using diploid data directly coming from DART-seq, we adopted the reads basically coming, the sequence reads coming from DART-seq. We took those and then created our own pipeline using different software, and then the variant calling using GATK basically for the variant calling in polyploids. And so this just kind of gives you an idea of where the bioinformatics component of this comes in. How am I doing on time? I'm not sure, I've lost track.
You're okay.
Okay, good. So I have a few more slides. So this would be basically be on the QTL mapping here, and we published this sometime last year. And so what we're doing now is going in depth into these sort of markers over here, or SNPs underlying the genome on chromosome 6, and looking at those in more detail and trying to see what kind of function would be related to any of those in terms of weevil resistance. This basically shows you the 11 chromosomes in banana, and these are typical kind of Manhattan plots, which show you kind of markers or SNPs in this case, which would be significant above this specific threshold, which have a significant effect for your trait of interest. And here we were looking at two kind of traits. One was looking at it. So we basically, for the weevil damage, we look at this in the culms of the banana plants, which is kind of at the base of the stem and the beginning of the root structure. And we did we would do a kind of transection where we sort of cut it horizontally, and the other one where we look at it in a vertical way. And so we do a kind of, so we call them, one was the TD, the transection, and the other one was the PD, the sort of more vertical cut in the stem. So in terms of how bioinformatics fits into the whole kind of supporting the banana breeding, we do a lot of sort of analysis of sequencing data, which which varies from RNA-seq data, what I've just shown you. We do a lot of marker discovery work, so discovery of variants and calling variants from whole genome resequencing data. So we have currently almost close to about 40 different varieties which we've resequenced. And I mentioned there's that project ongoing for about 100 different accessions. So from all that resequencing data, we look at subsets of that to call variants across the genome, and also using other skim sequencing technologies like GBS and more recently DART-seq. We support a lot of the molecular breeding activities. So that ranges from sort of analyzing the whole diversity within the banana breeding program or within the gene banks. So remember the CGIAR centers also hosts the global gene banks for a lot of these crops. And the banana gene bank sits in Belgium. It's called the ITC, the International Transit Center. And so we have a lot of material from there that has been genotyped, and we've carried out diversity analysis. We do a lot of sort of linkage and QTL mapping for some of those constraints of interest, genome-wide association studies for the more quantitative traits, and application of genomic selection using whole genome-wide markers to basically shorten the breeding cycle, so that we, rather than having the banana plants grow in the field for 2 years, when we get the tender leaves, we could do the genotyping of those and then do the selection based on the associated traits that we've identified earlier. So that saves us, a bunch of time and also a whole lot of cost in terms of growing these in the large areas for long periods of time. We also provide bioinformatics support to the genetic engineering and genome editing platforms. And in the newer projects, particularly probably not in banana, but in cassava, and this is something Michael is involved in, we're looking at exploring epigenetic variations. So when we look at the genome composition of a lot of these different varieties in banana, they're actually very close, but the phenotypes are very different. So morphologically, these are very different. Some of this comes because of sort of somoclonal variation when it goes through tissue culture, but also we feel that there should be some role that epigenetics plays in this. So whether that's methylation or other forms of sort of epigenetic variation. So in addition to sort of core bioinformatics, which is more kind of sequence analysis, we also support with our team here a lot of sort of what I would call breeding IT. So here we're looking at basically things like databases, so supporting BreedBase, in this case MusaBase, for tracking all the trial information, using electronic data capture, using barcoding in the fields to be able to label things properly and capture data on those particular things properly. And also we created some bespoke programs like, again, as I said, the crossing and seed set is a major problem. So we've developed this tool called BTrack, the banana breeding tracking tool. And here's a kind of screenshot of all the crosses that are done, when they were done, how much seed they produced, where did that seed go into tissue culture. So basically tracking it right from making of crosses to plants in the field, plants in the open field, and being able to track exactly what crosses were made where, and how those were followed up, and which plants are coming from where. So those things become important when you start applying genomics, because obviously the key to good QTL mapping, the key to good sort of GWAS studies is actually high quality phenotyping data. And so you need to be able to track your material properly, and you need to be able to have very high quality, accurate phenotyping data as well. And so we found that some of those were where the bottleneck was. And so we were also supporting sort of breeding IT infrastructure and software for these groups. So that's kind of in a nutshell what we do in relation to banana breeding. I hope I've been able to show you how what the constraints are in the breeding program and how genomics and bioinformatics can help overcome a lot of these constraints and help accelerate the breeding efforts of our teams here. Lots of people to acknowledge. I'm not going to name individuals, but we have a huge banana breeding team, the genetic engineering group, a lot of the transcriptome work we did in collaboration with New York University. And we've had several grants on the banana project from BMGF, the Bill and Melinda Gates Foundation, from the RTB, the root tuber banana groups in the CGIAR, and more recently through EIB, the Excellence in Breeding platform, and the Crops to End Hunger initiative. So I'd like to just acknowledge all my colleagues. This is obviously work from a lot of people in different teams, and also all the support from the funding agencies that we've had. So thanks very much. I hope I've kept to time, and I'm happy to take any questions.
Thank you so much, Trushar, for that amazing presentation. We have about 6 minutes for questions, and I can see some questions in the chat. So I can read them to you. The first one says, thank you for the interesting presentation. So what is the difference between genomic and marker-assisted selection?
Okay, so genomic selection is basically you're using markers across the whole genome. So basically you've got markers across the whole genome, and you're assigning them sort of effects or breeding values. So we call those GEBVs, genomic estimated breeding values. to each and every marker across the genome for quantitative traits. So if you have a trait like yield, which is controlled by a number of genes, which could be spread all over the genome, you would use sort of whole genome markers to do that. In terms of marker-assisted selection, that is where you've already identified, like, for example, we identified on chromosome 6 particular markers for weevil resistant that explain a percentage of the variation of that trait, in this case, weevil resistance. So we've already identified the specific markers linked to that gene, the causal mutation, if you want to call it, for weevil resistance. And so in that case, we would call that marker-assisted selection because you've identified specific markers, whereas in the other case, you're using markers across the whole genome. So I hope that is clear. I guess this question was from Michael Kitoy, but please feel free to comment if that's not clear. Yeah.
Okay, we actually have quite a number of questions. So the next one asks you to expound on the whole genome sequencing process, especially after the wet lab analysis is done.
Okay, not sure in which context, but the whole genome sequencing, there are two types of whole genome sequencing I should mention here that we do basically. And the wet lab process here, when you're doing whole genome sequencing, is basically taking your sample, extracting the DNA, and then creating libraries. So, and then, well, basically that DNA, you then share it, or you sort of break it down into fragments. And so that was the shotgun approach. And then those fragments are basically what you sequence by creating appropriate libraries. So when you sequence those, you basically get these short reads. And then there are two approaches. So one is de novo, where you actually assemble these short reads, but you need a lot of them. And so we say you need a high depth or a high coverage to do de novo sequencing. So this is where you don't have any prior information. But if you have prior information, like you have a reference genome available, then you can do what we call whole genome resequencing, where the short reads that you've generated, you then map those onto the reference genome available, and then look for variations in your sample versus the reference genome. So that's kind of what we do. I'm not sure if that answers your question, because you mentioned something about wet lab analysis, but I'm not sure what you're referring to there. But it's probably the library prep.
Sorry, I think you can comment in the chat.
Okay.
If that was clear or not. So then the same person asked, what is the future, describe the future of molecular breeding in Africa in the next 20 years.
Wow. Yeah, so I think it's a very bright future in terms of the application of molecular breeding. I think a lot of conventional breeding programs are now training people in sort of genomics and molecular breeding. There's also lots of new constraints coming with climate change, with new pests and diseases, with globalization. You've seen cases like the fall armyworm for maize, maize lethal necrosis. Now you're seeing in bananas the TR4. So there's lots of challenges that are going to come, which we hope that molecular breeding and the idea of knowing your genomes and the structure of those genomes better will actually aid in addressing quicker these problems. So more problems will arise, but we're basically developing tools and technologies to help speed them. So it's basically what you saw with, if you want to take COVID as an example, it's not that the COVID vaccine was developed within 1 year. It was information that they had from other viruses like SARS and other sort of respiratory syndrome viruses that they had already been working on for so long that resulted in the vaccine being developed within the short period which was kind of, you know, a sort of phenomenal feat in terms of application of research that has been going on, and then the whole regulatory process. So I think molecular breeding holds a really sort of key sort of cornerstone in the whole sort of feeding the ever increasing populations, but also in terms of addressing new challenges that come up, and improving yields, improving resistance, all of that. So I think it has a kind of bright future in that.
Thank you. Actually, our time is fast spent, and there are so many other questions. I don't know. I'd request you to, I don't know if you have the time to, we can post these questions in the HackMD, and then you can fill in the answers. Is that okay?
Yeah, that would be great. I'm happy to answer, and I'm also happy if, you know, the organizers can share my email, or I can put it on the chat. I'm just putting it on the chat. So if there's any specific questions that you need, yeah, I would.
Yeah. So Michael has shared the link to the HackMD. So the questions are going to be posted there, and you can just answer them there, and everybody can be able to access that.
Okay, I might not be able to do it right away, but within the next day, I should be able to find some time to just answer them. But can I have a quick look in terms of related issues in cassava can be resolved? Okay. What background checks do you carry out before releasing this plant, since those genes may result to expression of lethal genes? Okay, yeah, I'll go ahead and look at the HackMD.
Thank you so much, Trusha. Thank you for gracing us and for that wonderful presentation. I think at this point we can take a 10 minutes break, and then we come back with the next speaker. So we'll be back at 3:37. Thank
Great.
you, Trisha.
I'll use the break to answer those questions. So yeah, I'll get onto the HackMD now then.
Okay, thank you. Thank you,
Super.
guys.
Thanks very much. And thanks once again to all of you for all the good questions, and for the organizers for inviting me. So thanks very much.
Thank you for the introduction, Maggie. So sincerest apologies for this having come at this time. So obviously don't sign up to a conference with your Zoom government email, because this failed me massively, just didn't work. So I'll give a brief talk on pathogen genomics, and ideally concentrating on the theme of the conference. So looking at collaboration and community. So this essentially won't be specifically having to do with my work, but just giving an overview of what happens in the field and where you could see yourself as, depending on what your background is, and then just have a look at, you know, sort of the workforce that is involved in pathogen genomics and the research and etc. So we'll start with what is pathogen genomics. Again, just remember, I'm just giving a brief overview of the field. So this is not supposed to cover everything in its entirety with regards to pathogen genomics. So when you talk about a pathogen, so obviously you're thinking about a microorganism that, you know, causes or can cause some disease. And genomics, you're thinking about all these different aspects of genomes. So the structure, is it linear, is it circular? Where is a gene mapped on that particular part of the genome? What are the functions, and what are sort of the evolutionary dynamics of the different genomic regions or subgenomic regions? So pathogen genomics does marry the two. So you're thinking about pathogens, and you're also thinking about all these dynamics that relate to the genomes themselves. So, and I like this quote from this paper here. Yeah, that says that the genome of a pathogen at its highest possible resolution gives its identity and ancestry, the ways in which the pathogen infects us, and how it evades both antibiotic treatment and our immune system. So obviously here, antibiotic treatment could also include antiviral treatment, because when we talk about pathogens, these are not specifically limited to bacterial, but also, as we'll see, viral and plasmoidal, as well as other kinds of pathogens. So the one thing that is increasingly becoming clear is that when we talk about pathogen genomics, obviously we are not just focusing on humans, and realizing that, you know, we live in an environment that includes animals, and we also just have the environment. And these pathogens, a lot of them do circulate between humans and animals, and even the environment. And the idea here is that when you invest in protecting one aspect of this ecosystem, so whether you're protecting humans, in some way you do protect the animals and the environment as well. So the concept of One Health, the connectivity between the health of humans, the environment, and animals, is something that, you know, we can't ignore, and we'll have a look at some use cases of this, and particularly in SARS-CoV-2, the current pandemic, and how this makes lots of sense. So what is the utility of pathogen genomics? So again, this is just an overview, does not cover everything, but I often think one of the most critical bits has to do with diagnosis or detection. So in a sense, you always want to diagnose the pathogen that causes a particular disease. And often this would involve some aspect of a lab or wet lab component. So whether you're doing some extraction, then you do your sequencing, and be able to say that this is the pathogen that I think is causing this disease. Then the other bit, or which usually follows that, is that you're trying to investigate or characterize the pathogen that you've identified to cause a particular disease. So for example, you have the sequences that, you know, you've gotten out of the pathogen of interest. You're trying to, for example, taxonomically or phylogenetically place it in context with other pathogens, or probably the same species, or from the same genus but different species. And also you're trying to associate, probably we would say, what the genome characteristics and some of the symptomatics or the symptomatic patterns that you do see associated with that particular pathogen. The next step would be to track and describe transmission dynamics. So obviously, if you're dealing with an infectious disease, you know, that is transmitted either from one person to the next, or from one person to a given animal, you would also be interested in, or pathogen genomics can help you understand the transmission dynamics between individuals, between humans to animals, and whether you could be using phylogenetics or network models in that sense to be able to track the transmission dynamics and how that spreads over time. So both between individuals, and also, like, you could look at it also spatially, so how it's spreading from one location to the next. Last but not least, we use pathogen genomics to also find ways to mitigate against either infection or also just, also to treat once the pathogen has infected the given host of interest. So be it through the understanding of the pathogen itself, you come up with a vaccine, or you come up with an antiviral that, should be curative. And also some of these, based on the understanding of the pathogen, the mitigation strategies could involve neither of these, the first two, vaccine. And we've seen how effective some non-pharmaceutical public health interventions, such as just washing hands and wearing masks, have been quite effective against SARS-CoV-2. And also like secondary infections, things to do with suddenly a lot of diarrhea cases just went down because people just practice good hygiene. So ideally, I would categorize these four broad functions of pathogen genomics. And who does the pathogen genomics work? So I'm going to categorize this into four groups, and I like this outline given by CDC, the Advanced Molecular Detection Group. So one, we have laboratory scientists. So a lot of this would be involved in the pathogen detection and a bit of the characterization that we've talked about. So these are specialists who gather the samples, the data, do the sequencing, and be able to describe, for example, pathogen resistance to antimicrobials, and also to some degree, the spread of the pathogen in the population. And then we have bioinformaticians, who probably would constitute the majority of those who are attending this workshop. So a lot of this would use the data that comes off the sequencing machine, and in addition to computing platforms and software to integrate the sequence data with other metadata that's available. You're sort of trying to connect the dots, looking at the transmission dynamics, looking at the evolutionary aspects of the pathogen. So essentially you're trying to solve the outbreaks. And then we also have, on the other hand, epidemiologists. So they also have connecting the dot kind of role. So they would mostly be looking at how many cases are there, how are these cases, increasing over time or decreasing over time. And also, looking at the dynamic of spread in the community and integrating lots of metadata to understand the disease further. So while it might seem that, these roles are heavily siloed, so one statement I think I get a lot is you'd find a bioinformatician saying like, finally I've ran away from the lab. But essentially these roles are actually quite connected. So there's a lot of interaction between the different roles. And in the end, I don't think pathogen genomics as a field would work without collaboration. So it is the collaboration between the different roles that actually makes it work. So you cannot just be a bioinformatician, because obviously, if you told me that you went to GenBank and downloaded some data sets, somebody in the lab generated that data, yeah? If you're an epidemiologist, you're looking at this dynamics of community spread. I mean, somebody must have, collected that sample and told you that there's this pathogen in the community. Somebody must have probably sequenced that genome or done something related to, some bioinformatic analysis. So obviously, there is a connection between the workforce, and that's what makes it really a strong force to be able to understand all these dynamics related to these pathogens. And related to that, I'll just also highlight again 3 areas of research that are related to pathogen genomics. So, one of the areas would have to do with new technologies. So it could be in the lab. Scientists would always think of ways to target known and unknown pathogens. And so this could be designing better primers that would capture either the known or even the unknown pathogens that you think you might miss. Thinking about having more targets per run, so designing multiplex PCR, for example. Coming up with tests that have better reliability, accuracy, sensitivity, specificity, and predictive value. shorter turnaround times. So if you think about maybe doing an Illumina run, that would probably need a couple of up to 72 hours, or something like Nanopore that can start giving you results in real time. Again, each of them have their own disadvantages, but you know, you're thinking about definitely something, the shorter the turnaround time, the better, obviously. Cheaper diagnostics and even sequencing. So there's a lot of research that goes into this. I think the reason I'm trying to highlight this is that often if you are simply, you know, at the tail end of this thing, so you have your software on your computer and you're analyzing this sequence data, you might not be able to appreciate the kind of research that goes on into ensuring how that data comes to you. And a lot of this, there are people who've dedicated lots of research work, and actually most of them have feet in both places, so both in the bioinformatics and the wet lab research, to ensure that these new technologies actually do come out. So again, technologies that are more portable, if you think about it, then something like Nanopore, or even some of you know, field-based diagnostics that, you know, you could use non-PCR based in the field, you know, obviously also encompassing both wet lab and bioinformatics. And also, you know, thinking about new technologies for data sharing that is both safe and protective of patient data, and also coming up with data analysis strategies that are more efficient and collaborative as well. The other aspect that I think about has to do with surveillance. So this would encompass, you know, work that involves both epidemiologists and bioinformaticians as well. So you're thinking about tracking a disease progression and finding sources of outbreaks. And as I said in the beginning, so pathogen genomics research would involve different kinds of pathogens. So those who are in the bacterial field, viral or plasmodial field. The basic component here is, you know, all these have genomes, which, you know, you could use to track these pathogens. And as we said that, you know, we are increasingly appreciating the role of One Health. So thinking about both human and non-human hosts. And also, this is not just, you know, thinking about pathogen in a hospital setting, but you're thinking about vector-borne transmission. So you're thinking about foodborne infections, you're thinking about sexually transmitted infections, and those that you can just acquire from the environment as well. So different kinds of research that goes on in surveillance of all these different kinds of infections. And lastly, just looking at some of the interventions. So there would be lots of research that goes on in trying to identify new drugs, new vaccines, or better vaccines for that matter. If you've seen how the landscape has been changing for SARS-CoV-2, the need to update, you know, the existing vaccines to cover, you know, the newer variants. And again, just seeing the connection of how the surveillance, you know, and the interventions, you know, because without that bit of understanding what goes on in the community, you wouldn't know what to do with the vaccine itself. Thinking about controlling transmission. So this is thinking about both pharmaceutical and non-pharmaceutical interventions, depending on the pathogen. And also being able to research, you know, predicting the next outbreak. So whether you're using the current data to do some modeling in thinking about how you could predict where, you know, the next pandemic potential pathogen might come from. And also, so from which host, from which location, and how you might better mitigate that next pandemic. So I'll just give probably 3 use cases of pathogen genomics, and here gives an illustration of some of the epidemics and pandemics that have been there, or the major ones that have been there through history, you know, going back to the Neolithic age into the modern era. So somewhere here we would have the SARS-CoV-2, but lots of you, I mean, if you haven't read about them, I think you've seen movies about the plague and on the Black Death and things like that. So how has pathogen genomics been useful in investigating, understanding, and also mitigating some of these pandemics? So one, I think for me, one that in the modern era that comes quite quickly to mind has to do with the Ebola 2014. So this was a pandemic that had a case fatality rate of almost 50%. I'm sorry if the numbers are quite small, but ideally there were like almost 30,000 cases and about half of those actually died. And it was quite fatal, primarily primarily affected some 3 countries. So this would be Guinea, Liberia, and Sierra Leone. I mean, there are a few cases that were detected in Nigeria, Senegal, but a lot of those, like the transmission, it seems like the mitigation strategies against spread into those countries are quite effective. And here just showing the number of cases accumulating and the sequencing. So you could see that the lab bit was quite well done. A lot of these samples that were collected were actually sequenced. You can see this nice linear relationship. And these samples having been sequenced, the next thing that, that was done using bioinformatics techniques was to try and understand the transmission dynamics. So you might have come across this video. What it does show is that you can look at the cases here on the right. So as the cases increase, you can also see on the left the transmission from one location to another, both within country and also between countries. So using some very nice bioinformatics techniques, particularly phylogeography, to understand those transmission dynamics. The next thing that they ask themselves is, what could be the predictor of the spread that we see? So for example, when you see transmission between or within a country, is it something to do with the distance, for example? Is it something to do with, if you compare the destination and the origin of the transmission, if you compare the population densities or even the population size, if you look at things like to do with human movement. So you're putting in some component of mathematics there. And for example, in a generalized linear model, and asking yourself, what could be the predictor of this kind of spread that I see? So again, increasing the scope of what we would think bioinformatics actually does involve. So beyond looking at the sequence data, having a phylogenetic tree, and also doing some modeling work as well. And then that feeds into, you're looking at and asking the question, so we've had previous Ebola epidemics, so why did this one kill so many people? And you see particular mutations. So for example, this alanine to valine at position 82. And it was thought that probably this might have led to increased infectivity for this particular pandemic. And looking at going back to the lab and trying to assess the immunological or the reactivity of this pathogen with this particular mutation and the effect that it has on infectivity. And here you can see the A82V actually having this elevated effectivity compared to these other mutations. And even if when you incorporate these other mutations in addition to the 82, the mutation at position 82, you don't see any increased or elevated infectivity when you add this. And again, I mean, this information used to develop a vaccine that would be useful to prevent the next pandemic or even protect individuals in the ongoing epidemic. So here, just showing the scale of the use of both the lab, the bioinformatics, as well as the epidemiology in understanding the Ebola 2014 epidemic. I'll quickly go to COVID-19. So, which by now we have over 500 million cases, about 6 million deaths. And if you do remember, the first thing that came out was, the genome somewhere in early 2020, sometime I think in late January or early February. And with this, quickly, because the mRNA technology was available and was being used in other diseases, the vaccine was able to be designed, and immediately deployed. But then there were these questions with regards to where did the virus come from? And I apologize that this is a small image, but when they initially tried to look at different parts of the genome, they seem to have different origins. So you might look at one component, it seems to have come from a pangolin, and then you looked at a different part of the genome and it seems like it has come from a bat. And you looked at a different part of the genome and it looks like, it has come from a different bat species. So not the one that, you observed in this other part of the genome. So, and then you begin to think about things like recombination in these viruses and what that means, for the origins of this pathogen, and when, can we really say that we know where this pathogen did actually come from? And then obviously we've been trying to, looking at the transmission over time, both understanding how the different variants come about and how the spread goes on across between different countries and across continents. While that is happening, obviously, as you look at the different variants arising and also some, getting extinct from circulation, a lot of questions go back to like, can we come up, do we need to update the vaccine that we have? What kind of changes are we seeing, for example, in the spike? And if you've been following what happened to something like the Omicron, it had this many changes, and also like the question was like, do we still believe that these mutations arose from a human being, or was it something that was a result of a combination, or something that jumped from humans to another host and then came back to humans, with all these many changes? And related to that, sorry, I went the wrong way. SARS-CoV-2 now has been identified in so many non-human species, from household pets to animals in the zoos, to animals in the wild. And some both showed, to have been infected through natural infection as well as lab evidence. And like I sort of like intimated, the question is, when you see a new variant, for example, here in a human coming up, is it something that you could, probably infer as bidirectional transmission? So did it come from a human, or let's say to a mink, and then back to a human? And what might that mean when you think about, the next infection? So, and also when you think about, again, where did the pathogen come from? Do we really know? I mean, as the data really comes about. And obviously the other question you're asking is like, looking at all these dynamics, all this uncertainty, is this going to be a pandemic forever? Are we going to see a situation whereby, well, maybe it will die down and become a seasonal virus, similar to flu or the other seasonal coronaviruses? We really need an nth booster, which we don't know whether it will be the 50th or, just, we'll end up at the 4th or the 5th. Can we come up with a universal vaccine that, we wouldn't have to do, boosters? So something that would just be, it could be pan coronavirus vaccine that already some scientists are actually looking at. So this is also like another great use case for genomics. I thought I should also give something that is more closer home to Kenya. So a lot of the work that went on in our group was to look at transmission of a respiratory virus called RSV, in this case, looking at transmission within households. So here you have all these cases, collected from different individuals. And then, you've done your sequencing, you've mapped all the mutations that goes on in different genomes. And one of the questions that, we had was, if you look at the different members of the households, and if it's a household that has an infant, then who infects that infant? Should we assume that it's the mother? Because, the mother always spends the time with the child. Should we assume that it's a father? He always goes to work, at least in most African communities, then, might come with infections and infect the infant. Or is it, kids who go to school, or is it the visitors who come, checking on the child once the child is born? And then you use the power of genomics, so and epidemiology, and you're saying that I collected these samples at these different time frames, and I have these household members. If I generate a phylogenetic tree, what kind of clustering pattern do I see? And in some cases, you do see clustering patterns that you can't explain. And then, in such instances, you ask yourself, like, what else can I do to have a better understanding of these clustering patterns? And we had a colleague who would look at social contact patterns. So, for example, what he did here was to give different household members some devices that, when you come into close proximity, they would share data packets. And then this would give you a summary of how different household members interact. And for example, here you see that a lot of the interactions were structured by household. And then you would assess how the interactions occur by age per household, and also look at how the interactions occur by gender, for example. And, oops, sorry, one back. And also a colleague also looked at the modeling aspect. So essentially thinking that, okay, I know with certainty that this person showed up as positive on the 18th of February, but certainly this is not the date in which this person was infected, right? So if I'm interested in this person who had this test positive on the 22nd, might they have been infected by 4002 or 4004? And then you think about modeling, the dates before and after that might, if like the latency period when somebody is infected and when they might get infectious and things like that, and combining with all the genomic information. And then what we ended up getting from this work is that actually those who tended to infect the infant was more often than not, you have siblings who went to school because, they interact with all these different individuals, in schools and bring lots of different infections so often. But then also understanding that, the infant was also a culprit, often infecting the other household members. So because, as you can imagine, everyone is carrying the baby, is excited to hold the baby and things like that. So you could use pathogen genomics, so not just in a pandemic situation, but also trying to understand everyday infections in households. Lastly, hopefully we are good with time. So just giving an example of the kind of work that I'm interested in. So in my case, I'm trying to understand the seasonal coronaviruses. So we have what we would call the presumptive ancestors at the moment. And my question at this point, do these still stand out, or might this have changed with additional data that's available? And I'm using genomic and subgenomic data. So again, going back to the question that, do we have similar ancestral histories? If you look at the different components of the genome, when might they have emerged? Are they in a similar time frame or different time frames, and what might explain the different timeframes of emergence? Looking at the diversity, how diverse are they? What are the recombination patterns that goes on within and also between, the viruses themselves? And also something that I'm also quite interested in is doing ancestral reconstruction. So ideally, think about it, you have a present sequence and you're trying to work backwards and think about this sequence of this node, what might it have looked like? And then, you try to assess the changes that might have occurred in the history of that pathogen and what that might have meant. For example, if this was a bat and then this is human, so what changes are important in that adaptive process and things like that. And for that work, using all these kinds of different open source software and bioinformatics techniques. So, looking at recombination patterns, recombination rates, pairwise diversity, ancestral reconstruction, and phylogenetic analysis. So again, lots of open source software. And I must say that a lot of the recent successes that we've had in pathogen genomics has been attributed to increasing portability. So a lot of work going on, particularly with Nanopore, but Illumina as well. Decreased cost of sequencing, increased genomic capacity of sequencing platforms, so you can sequence a lot more samples in a given run. Improved bioinformatics capability, so both in terms of the software, the workflows, as well as the analysis, and also global platforms for data sharing and analysis, such as GISAID or NextTrain, ATIC network, always being very helpful when it comes to getting like the ATIC. You'll hear everybody talking about the ATIC protocol that has been used for a lot of the sequencing work for SARS-CoV-2. So what are some of the challenges that we do have in this field? So obviously there's not enough workforce, be it the laboratory component and bioinformatics and also the epidemiology. The sequencing costs are still high, particularly for the low and middle income countries, the bioinformatics tools and methods. So some of them, while it might seem that we have lots of these tools and methods available, there are quite some challenges, particularly integrating data from different sources. Data integration, obviously, is also one big challenge, and also data sharing. So both in terms of how to share it safely and also how to ensure that there's attribution for who shares the data. So I thought as one of the last slides to just talk about how do we build pathogen genomics in Kenya. So one of the things that obviously in some of our trainings that we're trying to emphasize on is trying to have local sequencing capacity. So moving away from the idea of having to ship samples into high income countries for the sequencing to be done there, having local laboratory bioinformatics and epidemiological expertise. So a lot of the analysis done locally. Having some, collaboration between individuals. So even within a country, so you might think, as an example, in our country. So those who are working in Kisumu, Nairobi, Kilifi, in Nakuru, different parts of the country. So that collaboration or networks that are established to ensure that this kind of work proceeds quite well. Open data, obviously, that's a key theme in this conference. And obviously the other component that always worries a lot of people is attribution. So somebody would want to share the work with you, but they would also appreciate that you attribute that output came actually from them. And also open source bioinformatics, so using software that is open source and also allows for a lot of reproducibility. So I'll just, the last 1 minute, highlight some of the work that we've been doing. So we've been doing lots of this genomic epidemiology trainings. So I'm based at the Fogarty, and we've been doing this with the Johns Hopkins University Applied Physics Lab. And we do lots of trainings using pre-recorded videos. So it means the participants can watch these videos at their own time. And then we also do have in-person lectures. And these are always followed by lots of interactions on Slack. So you could always ask a question, be it something related to the lab or bioinformatics or phylogenetics. And you can see that we've been training groups in Africa, Pakistan, in South America. So having all, and the other important thing is also like trying to encourage collaboration between members from a given country and also regionally, having networks within a given continent. So you can use, we have an open source software that is available and you could use to do lots of things. So check out this link from GitHub. It's open source. You can do lots of genomic and phylogenetic pipelines in there. It runs on your laptop. So we are trying to move away from cloud-based, in as much as it seems sometimes the nicest thing. But again, coming from a continent where internet is still quite expensive, this gives you a solution which you can run on your laptop, and anything from 4 gigabytes of RAM should be enough, and you can use this on Windows, Linux, or Mac. For hardcore bioinformaticians, so if you're a bioinformatics novice, it's no problem, obviously, click, 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 move on. If you know, like, oh, but I need to see what's under the hood, no problem. Definitely, you can go under the hood. So it's easy because these are just scripts run with an interface. So you can always tweak the parameters as much as you want. The automated updates, and obviously there's very good user support that you can always get. So I hope, as I end, have made a case for pathogen genomics, the utility of bioinformatics in pathogen genomics, and also the need for open data and collaboration in this field. So I'll be happy to take any questions. Thank you. Hello?
Thank you, James, for that presentation. There's already one question in the chat. So Irene says, thank you for the presentation. What's your view on the use of bacteriophages to combat AMR? Is it the solution we should focus more on? Are there ethical concerns surrounding the clinical use of these viruses?
That's a good question. So I did some work on phages, yes, but so I would say it's always, I think it's a viable solution. So I think already in places like Scandinavian countries, they are already using lots of gut reconstitution that comes from, you know, a lot of these phages, or, you know, phage cocktails as they call them. Obviously, there are always, you know, questions with regard to risks that are associated, and I think that also relates to, I think, an earlier question that I saw with regard to genetically modified bananas, for example. It's a good question to ask, but I would say that, you know, the fact that there are always risks should not prevent us from looking into whether these are possibilities of actually alleviating disease. So I think there is a lot of possibility of using phages in treatment. There's lots of work that's going on, and I think we can only hope that, you know, we get some good work out of, or some good solutions or mitigation strategies out of the phage work that's going on.
Thank you. Baseli asks, how do we access your trainings?
So currently, a lot of the work has been through, so two ways. So the ones that we do directly, we usually do proposals to the Department of State, and then they fund us to teach the countries and the groups that we propose to work on. And one of these should be Kenya coming soon. The other ways that we've done this is we've also gotten invitations from trainings that are organized by other individuals. So let's say the Bioinformatics Hub of Kenya wants to do a training on genomic epidemiology. You know, you could invite us to come as tutors and be able to be part of the training. So that's one way. So we do have a private channel which hosts the pre-recorded material. I think I'll just get that and share in the chat shortly once I get it. Then you can be able to view the videos and go through the training and, you know, download the software and even do this. So there's even test data that you can use to do this by yourself. Yeah. I don't think there are any more questions, right?
Yes, I don't see any more.
All right.
Unless somebody else has one more, I think we can take one more.
Yep.
Anybody?
Yeah, so I'll just put in my email also somewhere in the chat so that if anybody wants to reach out, they can always reach out.
Thank you, James. Benson, I think, says if you can show the link to the GitHub repository again, or put it in the chat.
All right.
Okay, thank you. So we'll move on to the next presenter, Joseph. Joseph will be presenting on Open Science in Africa project. And just a bit about him as he shares his screen is that Joseph is a graduate student at the University of Embu, pursuing a Master of Science degree in genetics. He's interested in molecular genetics and bioinformatics. Joseph, are you ready? Joseph, are you ready? I don't know if I can see him here. Okay, so maybe we can take just 5 minutes break as we sort this out. Can be back in the next 5 minutes.